Professor Jack Gilbert is a prominent researcher in microbiology and environmental science. His work has far-reaching implications for understanding the role of microbiomes in health and disease, including their potential relevance to conditions like Myhre syndrome. Professor Gilbert, we're excited to hear more about gut health. I've got lots of questions already, but over to you with your presentation. Thanks for joining us.
Thank you, Kate. I appreciate it. Let me just share my slides. So for everybody here, I'm a father of an autistic son, and have worked extensively with schools and organizations to try and develop strategies to help them work with children who are nonverbal and various other side effects associated with some of the neurological and developmental disorders that come along with neurodiversity. But in the context of what Kate asked me to talk about, I wanted to give a broad assessment of the microbiome, the bacteria inside your body, which I've spent the last 20 years working on, and examining how they associate with gut health. And a lot of this is in part due to my own experience of trying to establish a healthy gut microbial community and a healthy repartee between the microbiome and the body in my son and myself. Hopefully you'll find it useful, even if it's not directly associated with Myhre syndrome. So in the greater scheme of things, the first thing I'd like to say is that you need to examine the microbiome in your body in the context of the immune system, right? We have evolved in a sea of microorganisms. All of the species you can see with your eyes have done so. And that relationship between the microbiome and everything you can see with your eyes has evolved a necessitated mechanism to handle that exposure, right? Microbes are just living organisms that are trying to find an environment in which to replicate in order to spread their genetic material, right? Just the same as we are. And we are a very nice, warm, nutritious, rich environment. And so over the years, we evolved a mechanism of managing that microbial exposure that we all get. Each one of you right now is emitting about 30 million bacteria into your immediate vicinity every hour, right? We are literally, think that kid from the Peanuts cartoon, right, who has his blanket and is just shedding microbes everywhere. Pigpen, that's him. So you can imagine Pigpen walking along, spreading microbes. You're doing the same thing. And so every time you're interacting with somebody, you're picking up some of their microbes and shedding some of your own, right? And anytime you walk into any environment, be it outside or inside, you're being exposed to new microorganisms. The same is true for a child or a baby or anyone. So the immune system, less like an army, is more like a national park service. It's there to manage the diversity of microorganisms that we're exposed to explore how they interact with our body and maintain those good interactions which we need to keep us healthy. So, you know, your gut is like the inside of a donut, right? It's connected to the outside on two ends. And inside your gut, your immune system is managing that microbial garden to keep the good crops growing and weed out the weeds. And so the immune system has become vital as a surveillance network and a national park warden. Now, when we actually understand and examine the microbiome in the instance of health, we have to consider two instances. And I'm using two extremes here, but I use them to illustrate the two sides of the coin of microbial dysbiosis. So what happens in a healthy gut? So this is a cross-section of a gastrointestinal tract here on the left. This is the membrane that lines your gastrointestinal tract. These little cells are called colonocytes with their little fingers sticking up into the gut environment, and they're there to absorb nutrients and sugars and fats and carbohydrates, etc. And inside the gastrointestinal cavity, it's mostly anaerobic. That's represented by the blue color here. And these blue bacteria are ones we call obligate anaerobic bacteria. These are very, very good at fermenting or digesting the fiber which you cannot digest, right? We all heard about fiber and the need of fiber in our diet. The majority of the fiber we can't absorb, but these bacteria can. And they break down that fiber, and in doing so, release chemicals called short-chain fatty acids. These short-chain fatty acids here, such as butyrate, are really, really good at absorbing into these cells. And these cells have evolved to use butyrate as a preferential food source. So for over the eons of our evolution as a mammal species, the bacteria that have degraded fiber through all of the ancestors of our species have also caused the evolution of our body to become dependent upon those chemicals. So the bacteria degrade the fiber, they release this compound butyrate as almost an excretion product, and our cells that line our gut use that to fuel their growth and their maturity. This leads to a mature, strong barrier separating all of the microbial life and food and gubbins inside your intestine from the rest of your body. And these bugs, these cells that line your gut are also extremely good at absorbing oxygen because they're growing and they're healthy, right? So they suck up the oxygen here, stopping the oxygen from getting into the gastrointestinal tract, keeping it anaerobic, thereby promoting the growth of these obligate anaerobic bacteria, these bacteria that hate oxygen. These short-chain fatty acids are also fuel for your immune system. They keep your immune system ticking over, especially these T regulatory cells, which are incredibly important in driving down inflammation. So a healthy gut has these bacteria living inside it, which are promoted by the immune system, are kept in the perfect environment by an oxygen absorbing layer. And they're also providing fuel for both the cells that line our gut to keep that barrier strong and the immune system to keep inflammation down. In an unhealthy gut, or what we refer to as dysbiosis, that relationship between the microbes in the body is broken down. Let's say you've eaten an unhealthy diet for a long period of time, or you've taken an antibiotic for a short period of time. You're going to knock out some of these healthy obligate anaerobic bacteria, and you're going to let some of these facultative anaerobic bacteria proliferate in their absence. And these bugs, such as E. coli, Klebsiella, Salmonella, which we hear about and we know about from disease-causing organisms, foodborne pathogens, are also present in your gut, and they're very, very good at growing. And they release compounds like hydrogen sulfide, that rotten egg smell, or flagellin, which is what makes up their little tails, or things like lipopolysaccharide, which is a product of their membrane, which are all potent stimulators of inflammation. Your immune system sees these chemicals and it goes into overdrive trying to tackle their presence. Also, these bugs don't digest fiber, they don't release any short-chain fatty acids, and therefore these cells which line your gut go into anaerobic glycolysis because they're not using the right fuel source. Oxygen leaks into the gastrointestinal cavity, further stopping these obligate anaerobic bacteria from growing back and keeping this gut environment immature and also permeable. So now you've got a leaky gut where things can get from the inside of the gut to the body. So this relationship is a breakdown. It's a negative feedback cycle. And the only way to really overpopulate that is to feed the good bugs with as many good chemicals as possible in your food. So in your fiber and polyphenols, etc. So what does a dysbiotic environment mean? Well, it can mean many things, but we know it's associated with obesity. So in this study we did a few years ago now, we took germ-free animals and we exposed them to these are germ-free animals, these mice have no bacteria at all, they're completely clean. Think Boy in the Bubble from the 1970s kind of idea. They've never had a microbial exposure. We take these animals and we give them a low-fat, high-fiber, high-polyphenol, perfect diet, or a high-fat, high-sugar diet, and we pump it into these animals at exactly the same calorific intake. Because there's no microbes in the gut, we see no disruption in circadian activity between the brain and the body, and these animals stay lean or obese depending on how many calories you feed them. So the perfect diet has no impact on obesity if you don't have bacteria. But if we have bacteria, then a low-fat, high-fiber, high-polyphenol diet, such as the Mediterranean diet, will lead to what we refer to as normal microbial oscillations, where this diet will promote the bugs producing short-chain fatty acids, and we'll get normal microbial signals, which lead to normal circadian function in the brain. The kind of circadian function most of us are feeling all the time, where our brain and our body are in sync. You can think of disrupted circadian function as what happens when you travel from, say, here to Europe or here to China or India, where you suddenly feel like your brain and your body are out of sync. You might need to poop at the wrong time of day. You might get hungry at the wrong time of night. Your body isn't in sync with your brain. But a low fiber, the high fiber, low fat diet can lead to a lean phenotype, even in the presence of microbes. But when we feed that same mouse a high fat, high sugar diet, we get very aberrant microbial oscillations, generally punctuated by some of those facultative anaerobic bacteria with the flagellin and the hydrogen sulfide. These chemicals get into the bloodstream and they can cause our body to sense time differently to our brains. And this can lead to obesity. Again, exactly the same thing happens with jet lag. If you're jet lagged, your body and your brain are syncing thinking time differently, and this can lead to an increase in fat mass accumulation for the same calorific intake. So a high fat, high sugar diet is only effective at promoting obesity in the presence of microbes. Of course, if you eat a high calorie diet for any of these, right, then you're going to put on weight. The more calories you consume, the more weight you put on. But in the presence of microorganisms, this high fat, high fiber diet can create obesity even with a low calorific intake. We also know that this affects things like surgery. So in this context, I'm going to take a piece of damaged gut tissue, and I'm going to remove it, what we call a colonic resection. And in this case, I let oxygen into the gastrointestinal cavity, which kills off our oxygen-hating bugs, the good bugs that ferment our fiber. I then pump the body full of antibiotics, which kills off everything else. I then stitch the gut back up, forming an anastomotic junction here. And the body then goes into overdrive to repair itself. And it actually sucks nutrients out of the gastrointestinal tract and causes the bacteria in there to go on a scavenging mission, trying to find the best kinds of nutrients. In doing so, they form what we refer to as a biofilm. They grow on the wound site, start to break down the collagen that connects the tissues in our gut and allows the gut contents to spill out by opening up the wound. This is called an anastomotic leak. What's happening here is that bacteria present in the intestine before surgery, promoted by a high fat, high sugar diet, as well as other lifestyle factors like whether you have furry pets or no furry pets, or whether you live in a highly green environment or a very urban, polluted environment. All of these can lead to the presence of bacteria inside the gut that when we go through the multiple insults of surgery can become highly abundant and can promote a wound damaging environment in post-surgical recovery. So we've got bacteria living inside our body that are promoted in growth by the wrong kind of lifestyle that can even affect how our body will respond to surgical theatre and practice. And then we did a really interesting experiment. This is a very complicated figure, so don't worry about the details on the left, but focus on these on the right. We took mice, we gave them either a Western diet or, like a high fat, high sugar American diet, part of my accent. But we gave them a very high fat, high sugar diet, and we put them through surgery, or we gave them a high fat, high sugar diet, and we added one of these bacteria in that likes to break down the collagen matrix of your intestine when it's under stress, like we saw before. So here I've got these collagenolithic bacteria that love eating collagen, and I've got colorectal cancer cells, and I put them inside this animal. If I give this figure here, this figure C down here in the middle, the black and white figure, the most boring one on the page, is also the most informative, right? If I give this mouse a chow diet that's high in fiber, high in polyphenol, and I give it antibiotics and surgery, the chances of local tumor formation in that animal are very, very low. If I give it a high chow, so high fiber, high polyphenol diet, plus antibiotics and surgery, plus the presence of a collagenolithic bacteria, in this case, Enterococcus facialis, my chance of getting local tumor formation or the development of cancer goes up. If I give the animal a Western diet plus surgery, the chances of cancer formation goes up dramatically, right? So a Western diet is very pro-cancer generation in the presence of cancerous cells. But if I give it the same treatment, so Western diet, I give it antibiotics and surgery, and I give it this collagenolithic bacteria, which can damage the matrix of the gut, I get a significant increase. Almost 90% of our animals develop severe cancer, gastrointestinal cancer, and will die. So this high fiber chow reduces the presence of these collagenolitic bacteria and the effectiveness of these collagenolitic bacteria, and alters the chance of tumor formation. So we're talking about developing targeted prebiotic and probiotic formations, in this case, a prebiotic called phosphate, which seems to inhibit the activity of bacteria, to actually reduce the risk of colorectal cancer formation and also surgical damage inside the gut, but also potentially to just create a healthy gastrointestinal environment in these in people. We also know, and this is relevant to both the treatment or the help that we give children with autism or Myhre syndrome, we know that the microbiome is associated with cognitive and neurodevelopmental activity, neurodegenerative disorders, even mood and behavior like depression and anxiety. We also know that most neurological abnormalities or neurodiversity can also be associated with GI disorders, so gastrointestinal disorders like diarrhea or constipation. I know my own son had both conditions very early on in his life, up until about the age of 7, and we struggled with that a lot. We also know that things like antibiotics are actually linked to behavioral phenotypes. So when you take an antibiotic that knocks out bacteria, depression and anxiety are significantly more likely in an individual, as well as behavioral abnormalities, such as loss of speech, cognitive performance, and other factors that we traditionally associate with autism. But interestingly, antivirals and antifungals don't seem to have any effect whatsoever upon neurological activity, suggesting that it's the presence of certain types of bacteria in the intestine which is linked to some of these abnormal behaviors. We also know that we can take the gut bacteria present in clinically depressed people, to take their poop, right? Take their poop, make it into a poop smoothie, and allow or force that into the intestines of mice, and those mice will show depressive-like behaviors. Whereas if you take the same kind of poop slurry from people who are not depressed and you put it into mice, they show no significant behavioral change, suggesting that it's the presence of certain bacteria or the absence of certain bacteria in the gut of people with neurological symptoms which can elicit that change. We know that this is done in 3 ways. This figure looks complicated, but it's really not. Bacteria in the gut produce neurotransmitters like GABA, norepinephrine, dopamine, serotonin, the precursors at least of, and they drive tryptophan metabolism inside the body. So significant amounts of neurotransmitters are produced by bacteria in the gut. We also know that they obviously regulate immune inflammation. I mentioned this before with those short-chain fatty acids driving T-cell accumulation. So they can either drive down inflammation, or they can promote inflammation with things like hydrogen sulfide production and lipopolysaccharide production. And these immune cells circulate throughout the body, driving a cytokine reactivity, which can cause neuroinflammation, which is often associated with aberrant behavior, such as outbursts, anger, violence, depression, and anxiety. We also know that neurological links between the gut and the brain are paramount, such as the vagus nerve, which links gut activity to brain activity. People used to have their vagus nerve chopped in order to stop the generation of peptic ulcers, right? So we used to have that. It proved that it was actually not effective at alleviating peptic ulcers, so people have stopped using that as a strategy. But we still know it carries nerve signals. And then spinal pathways, so the gut is connected to the brain through nerve endings which go up into the brain. And we explored this phenomenon by looking at rats, because rats are actually better models of human neurodevelopment and human activity than mice are. Mice are very bad models of this. So here we took what we refer to as a Sprague-Dawley or SD rat, which shows very limited neurodevelopmental or depression symptoms. They appear to be what we refer to as healthy controls. And then we compared them to fawn-hooded rats, which are a depression model. Fawn-hooded rats show a lot of anxiety, a lot of depression. They're a type of rat that is bred to represent a depressive phenotype. So we took feces from Sprague-Dawley rats and gave them to fawn-hooded rats. Remember, rats are involved in coprophagy. So here, poop eating. So here, I've got a number of different tests. I've got a forced swim test, an open field test, and a sucrose preference test. And in all cases, I've got the FH rat, which shows a higher score on our forced swim test suggesting depression, a lower score on our open field test suggesting depression, and a very low score on our sucrose preference test. They show what we refer to as anhedonia. If you are hedonistic, you might love sugar and you'll eat a lot of it, right? But it also makes you pretty happy. Whereas animals that are anhedonic, they don't like, they're not particularly happy and they don't like sugar. So, and we can see in all cases, the SD rat here in pink is opposite in its trend to the FH rat. But if I give the FH rat poop from an SD animal, we'll see that the opposite is true. It starts to look a lot more like the SD animal originally in terms of its behavioral tests. Interestingly, when we look in serum, we don't really see any significant effect of the microbiome on serum levels of, say, serotonin. But when we look in the hippocampus and we look at the serotonin and norepinephrine levels there, we see significant changes in the microbial, in the levels of these neurotransmitters when we take an animal, an FH animal, and we give it an SD microbiome. We see significant changes, much more similar to what we would see in our baseline SD animal. So this suggests that you can rescue this phenotype of depression by transplanting feces from one animal to another. What we think is happening here is actually due to the metabolites or the chemicals these bacteria produce in your gut and how they get into your brain. So in our FH rat, we see really, really high carbon-rich level metabolites. So these are metabolites coming out of the bacteria's metabolism. They get into the bloodstream and those get into the brain, and they appear to drive changes in brain behavior. But when we give the same animal an SD microbiome, the microbiome from an SD rat, we show a change in microbial diversity, an alteration in the level of carbon metabolism, and an increase in nitrogen metabolism by these bacteria, which leads to an increase in nitrogen-rich metabolites in the bloodstream. These get into the brain, and we can see them elevated in the brain. And this appears to drive down hippocampal osmolytes. Brain inflammation goes down significantly, and the amount of neurotransmitter production goes up, especially in the hippocampus. So this suggests that the gut metabolism, gut bacteria, and therefore diet is significantly linked to what goes on in the brain. And we've shown this with humans. So we took humans and we gave them a fMRI scan, a functional magnetic resonance imaging scan on people with major depressive disorder. And we used a particular association, a link between these little blue bits here in the brain, which suggest a altered GABA-mediated response of prefrontal cortex connectivity. And then we associated that with the change in the bacterial contents of our intestine. So here, the abundance of Bacteroides in the gut is negatively correlated with brain signatures of depression inside the brain. So we hypothesized that this could be due to the fact that the Bacteroides were actually synthesizing gamma-aminobutyric acid, or GABA, in the gut, and maybe that was influencing brain activity. So to explore this further, we took one of these bugs and we started a company called Microbiome, of which I do hold a very small percentage of equity, which I believe is worth about $3. Apparently I'm not very clever and not in this for the money, but we want to see if we can commercialize this product. We can show that this Hollow-1 bacteria, this Bacteroides organism, is very, very good at alleviating symptoms of depression in rats. So in here, I got a sleep-deprived rat, and it shows a very significant increase in its time being immobile. So it's very, very depressed. It's very mentally fatigued. If I give it this bacteria, this Hollow-1 bacteria, it significantly reduces the time it is immobile, much more like the control, even better than the control in some cases, suggesting it creates more alertness. We also know that it has a very potent anti-inflammatory effect, and that it's correlated with lower inflammation when it's detected in people. So people have lower levels of inflammation when you detect this particular organism in the gut. So we've taken this organism and we're running through with a series of models. So here we take animals that are stressed and we give them particular treatments to alleviate their stressed attitude, such as ketamine. Everyone might have heard of ketamine as an antidepressant, and it does indeed rescue this phenotype of sucrose preference. Remember I said animals that drink more sugar tend to be happier, those that don't tend to be sadder. So we have stressed animals not consuming as much sucrose, But if we're giving them ketamine, their sucrose preference goes up, so their symptom of depression is alleviated. But we get the same level of depression relief with just augmenting it with this probiotic bacteria. That's a phenomenal change, right? So instead of trying to hijack the chemical activity of the body, we're just adding a bacteria back into the animal's intestine, which it's missing, and which people with depression, anxiety, or neurodevelopmental issues may also be missing. So what are we doing? So in the last few minutes, what are we doing? Well, we are examining the microbial community of our body. So if you are, I'm assuming most of the parents here will be familiar with this chart. It's called the Bristol stool chart. And we go from everything like rabbit droppings to gravy, and everything in between. When you look at somebody's poop, you can tell a lot about this. People have been doing this for years, using people pooping in a chamber pot, and your physician will look at your poop and say something about your health. All we're doing is taking genetic information out of that poop and using it to say a lot more. So we call this microbiome-wide association studies. These are akin to what you may have heard to as a genome-wide association study, or a GWAS, where people look at the genes for obesity, or the genes for depression, or even the genes for autism, which have never really been proven. And we try and identify how they are distributed in a population and whether they associate with disease. We do the same things with microbes and the metabolites they produce. You know, these metabolites are interesting. So bacteria in the gut can actually augment or change acetaminophen inside that you consume in Tylenol, for example, and make it into acetaminophen sulfate, which can damage your liver. So you have the wrong kind of bacteria in your gut. Consuming acetaminophen on a regular basis could actually cause liver cirrhosis. We also know they can break down the chemicals which our body produces, like bile acids, and make secondary bile acids, which can actually protect our body from infectious disease. So we've been doing this in something called the American Gut Project, where we've surveyed over 30,000 people's microbiome profiles. And we've used this information to build up a repertoire of understanding about the microbial diversity of our bodies. But we've actually failed to miss on one particular important piece, that the gut microbiome changes over time. So here I've got on the x-axis the number of time point samples, the number of consecutive poops collected from an individual, and my ability on the y-axis to identify a biomarker or a particular bacteria or chemical that's associated with disease goes up significantly with the more samples I actually acquire. Why is this so important? Well, unlike things like height, weight, and age, which don't really change in the small numbers, or blood pressure and insulin, which are very small numbers but change every day or even every hour, or genetics, which is lots of genes and high dimensional. The microbiome is like all of these things. It's very high dimensional, therefore it requires big numbers of people to account for dimensionality, or frequent sampling to account for dynamic changes in the microbial system. In a disease or a disorder, or even a neurodivergent activity, this can be difficult because we hard getting the large numbers of people, but we can get large numbers of poop samples. So to facilitate that, we built something called the Gut Lab, which is being trialed in a $200 million initiative I'm leading for the NIH right now, where we are trying to understand how people respond differently to different diets. We put this device in people's bathrooms. They enter a PIN code in the top. They drop a piece of used toilet tissue in the top of the device, and it extracts all the DNA and the RNA and the metabolites from that poop sample and stores it for downstream analysis. This is really, really important because it can help us as scientists to track every single stool sample somebody produces and use this information to change our understanding of health in people. So we're doing this to create personalized prediction of nutrition, right? This is because depending on who you are and the type of food you eat will have a very big and different impact upon the types of nutrients you're consuming. So this can be vitally important in helping us to understand, help us understand how things are changing in our body. So people that eat ice cream might have different responses. People who eat white rice might have different responses. And so our new initiative is focused around this, and we're launching our ability to predict how individuals respond to food and dietary patterns in our bodies, including people with neurodivergent attitudes. So what can a microbiome do for us? Well, it can help us to develop diagnostics to predict diseases. It can help us to identify how microbes drive molecular dynamics in the body, which can be used to treat things like allergic, metabolic, or neurological disease. It can even help us to understand how to use antibiotics more effectively. It can help us to redefine how we interact with our environments. You know, think about the hygiene hypothesis and living on a farm and the benefits of doing that versus living in an urban environment. It can help us to understand how to design clinical trials to get at high-risk diseases in populations, and even help us to create new technologies and informatics to support the globalization of diagnostic and therapeutic potential. So thank you to, I can't show all of my donors here, but for some of the people that support this science, and to my lab. Thank you, and I'm happy to take questions.
Great. Oh, thank you, Jack. That's incredible. We've not had a talk on this subject matter before, so I think people are just, excuse the pun, digesting everything that you've been talking about. But I've got some questions here for you. Constipation. So our community experiences this, and even with a good diet and using things like Miralax or lactulose, they can still have problems. With children, as you've mentioned, there are other psychological factors to add into the mix. We also have many members of our community with an ASD diagnosis. We talked about this personally. I guess just thinking about all of those factors, any advice or how to approach trying to get some good gut health going in that situation?
Yeah, I mean, I will drop any illusions as to the ability to get children with autism, and I'm assuming the similar situation might occur in Murray syndrome. Get children to eat a predominantly healthy diet it's very difficult to get most human beings to develop a healthy diet. You know, people generally in adults, they don't tend to want to eat healthily. They tend to want to eat pretty unhealthily because unhealthy tastes better and is easier, right? And often a lot cheaper, especially if you're in the lower socioeconomic bracket. However, there are ways of getting around that. So, for example, with my son, we explored, after working on a few clinical trials, the deployment of butyrate directly. So remember I mentioned the bacteria ferment fiber in your diet, and then they release this chemical called butyrate. So we figured out a way of working with a chemist to get the butyrate into a form which could be acceptable to a child, because butyrate smells like vomit and tastes like vomit. It's not a nice thing to consume. You don't normally taste it because the bacteria produce it in your intestine. And so so if you can get butyrate into the diet, it can have anti-inflammatory effects, and it did seem to promote a shift in neurological activity. There's also a number of different fecal microbiome transplant strategies which have been implemented in trials, where taking feces from a neurotypical sibling or parent into a child's intestine has shown a significant improvement in symptoms of gastrointestinal distress, and also in some neurocognitive performance, such as speech and a reduction in outbursts. But we're not there yet, and the FDA does not regulate or approve the use of fecal microbiome transplant for treatment of neurological disorders. So these things are really difficult. I just noticed in the chat that Christina Hazard suggested recommending a nutritional consultation. I do, but unfortunately I do because I think it could be valuable and it could give you some tools. But unfortunately, most nutritionists are using data to suggest nutritional advice that's based upon a series of tests that were done on white male college students in the 1970s as to how they responded to diet. This is the reason the NIH is finally investing some money in food as medicine, and our new $170 to $200 million program that we just launched is a phalanx into the ranks of ignorance that we have around the importance of nutrition in our lives, right? And so the best thing I think you can possibly do is to try and reduce the amount of simple sugars in a diet, try and increase the amount of colored fruits and vegetables in a diet, even if in smoothies. My son loves smoothies, always has done, right? Some kids don't, but he has, and they're very effective means of getting that nutrition in there. And then trying to promote fermented foods can be really valuable if you can get them to consume fermented foods, or just a higher fiber diet can be valuable. And again, if all else fails and they still want to eat hot dogs that have no taste or coloring, then nutritional supplements can be a good stalwart against effective treatment. But yeah, I will not give any pretenses. It is very hard to get kids to eat healthy. They just don't tend to do it generally, and unless you can figure out a good way, I'd suggest. I did actually roll up. My son's now nearly 17, and he's in culinary classes at school, and he's loving it. But I don't think that, I'm sure they're teaching them culinary skills, but most of the things he makes are pizzas and baked potatoes and mashed potatoes. And I'm like, I think you're missing the point here, but it's all good.
Doing something though, he's cooking. He can make you a jacket potato. You can add the salad. You can add the salad. I've got a question here. If people have to use Miralax or lactulose, how is that impacting the intestine? And then I guess, therefore, how do we try and compensate for that?
Yeah, I mean, sometimes it's necessary, right? That's one of the side effects of any kind of central nervous system disorder is constipation. Your body actually starts to alter the peristaltic regulation in the gut in ways that we don't fundamentally understand, but it appears to be related to a shutting down of bacterial metabolism of key compounds like GABA. And so promoting that is good, right, in any ways to do. So even if you have to take Miralax or lactulose, if you can get a fiber-rich, polyphenol-rich drink or food into the child before and after that, not forced, but into their diet rather than just having them eat hot dogs and chips, then it can help, right? It can improve it. The actual intestine itself is not dramatically influenced by lactulose or Miralax or any other kind of mechanism to loosen the bowel. But what it does do is it flushes out the gut. Now, it doesn't get rid of all the bacteria. All the bacteria are still present in the intestine. So the best thing you can do is feed those bacteria the right kinds of food to promote that homeostatic, that healthy gut microbiome inside the gut after you've used or during the use of say Miralax product.
Okay, thank you. I met you at a conference earlier this year, and we talked about microbiome research and what that might look like for communities like ours. So can you give an indication of how communities like us could get involved? Would it be that we add a stool sample to clinics when we visit clinics, or would it involve one day ideally having a sample set of the device you mentioned in the home? What's the vision for how we actually try and gather some community data?
Well, I think you and I have talked about this as well. The problem with Murray syndrome particularly is it's quite small. So it would need to be, it's a rarer disorder, right? So it would need to be associated with a larger clinical investigation as a subset cohort. I use those phrases because that's how the medical community talks, right? We subdivide everybody and we depersonalize them to try and create knowledge. But at the end of the day, it's my son in those trials. But the framework that we need to think about is how can the community itself come together to try and understand and identify the issues which the children and adults are facing, right? Is it possible to create a series of common phenotypes, common parameters which the children might be feeling, like differences or food allergies, food sensitivities, activity sensitivities, alterations in speech activity, neuropresentation, writing everything down, creating data libraries, data repositories within your community can be highly effective in getting communities of researchers interested or involved in those practices. If you can say, hey, we are parents, we're struggling with this situation, but we're also interested in gathering as much data as possible to help us communicate the commonalities between our children in this condition, I think that's vital. And obviously with a condition like Murray syndrome, which has a diagnosis, it's possible to start to do that at scale. The other piece is, there are a lot of companies out there that will sequence your gut microbiome, but they won't necessarily provide you, well, they cannot, in this case especially, provide you with any relevant information. We just did and published the largest meta-analysis of autism spectrum disorder based upon a large number of research programs, most of which I ran over the last 10 years to try and understand how the microbiome differs for children with autism versus not. And really all it proved was that there are such a great level of diversity across the spectrum, right, of autism, that it's virtually impossible to identify a single feature which could be augmented or changed or removed in order to improve health. So I think we do need to work on N of 1 studies. We do need to say that work on trying to figure out what strategies could help my child and figure out what the strategies, how to implement those strategies. And then you can build up an understanding of whether it works and whether it doesn't based upon adding it and then removing it, whatever it is. We did this with butyrate supplementation, and it was when we removed it, my son's condition got worse. His quality of life got worse. And then when we added it back in, his quality of life got better. My wife didn't like me continually doing that 3 times, but it was effective at providing us with the peace of mind that what we were doing was actually effective, right? And wasn't just changes in the developmental profile of my child. Yeah. So it's complicated, right? But I'd say as a community, you have to come together and continually advocate for better understanding of the condition within the population and within the National Health Institutes of Health.
Yeah, that's really good advice. Thank you. One final thought, because you've given us some great things to take away in terms of the practical side and things, but it was interesting that you talked about more of like the holistic approach, and that the neurological factors, the vagus nerve you talked about. Are there other holistic things we can do other than, the high fiber, where you've said there can sometimes probably be some resistance there with our children? Like, holistically speaking, can we think about our microbiome in that way, and what kinds of things does that mean we can put into our lifestyle?
Yeah, I mean, the basic things are small changes. Like, there are prebiotics which you can take, which can, if food is more complicated, whole foods are more complicated to consume, then identifying, and if they can swallow pills, not everyone can swallow pills, or if they can consume powders, right? There's the possibility of adding prebiotic supplements to an existing diet to try and fortify that diet. Holistically, there's also cognitive behavioral therapies which can be highly effective, right, in supporting a child's development, in allowing them to understand strategic processing. Again, my son's 16, 16 and a half, and he's learning to drive, which I never thought would be possible, right? But every single day, we work with him to try and figure out what he needs, and I think that's the N of 1 ideology. So at a holistic level, compassion and support within a community, within a family, and with the school. I really did like the last talk, because I had to use advocates to get the treatment that we needed and the support that we needed for our IEP in school. Those advocates were incredibly valuable at helping us to understand the legal frameworks, especially when we moved to schools. So they can be incredibly useful. So strongly advocate for advocates. But so a supportive system in place on top of a proper nutritional regimen is highly effective.